All right, it is 12:30. So we'll get started. Grades for test one are up. If you haven't seen that yet, you should be able to go back and look at test one. Instead of spending time in class, like going over test one question by question, what I'm going to do is make a little video guide going over just some things on test one. In case you got something wrong, not sure why. And so I'm planning on doing that sometime in the afternoon. As always, if you have questions about how I graded something, you want to disagree, feel free to email me and we can either set up a time to talk. If you wanted to go over that, it'd be easier to express your opinions that way, or we can handle it through email. Either way is fine with me. I have applied the extra credit to it. So the score under test one grade should be the score for the test. Remember, I do drop your lowest test grade. So if you didn't do so well, especially with like everything that happened the week leading up to it, don't worry, it's not going to kill your grade as long as you continue strong from here on out. All right, so moving on Thursday, we started to talk about the citric acid cycle. And we ended our discussion about the first complex in the citric acid cycle, the pyruvate dehydrogenase complex. So this is what will take our pyruvate from the end of glycolysis and will create acetyl-CoA for us. This is irreversible, and it's once you go down this pathway, you are committed to becoming acetyl-CoA. And we talked about the 5 different steps of that for this enzyme. And the very last thing we did was just review the mechanism. So that was Thursday. So we just have a few more things to cover about the PDH complex. And one of those is how does our substrate move between our different enzymes? So if you remember, PDH complex, pyruvate dehydrogenase complex, is made out of 3 enzymes, E1, E2, E3. That's what's being shown here down in the pictures. These greens are E2, I believe. I believe this is E1. I believe that's E3. And so you have this giant multi-protein complex where all the proteins are right next to each other. All the enzymes, rather, are right next to each other. And so to facilitate the movement of our 2-carbon unit, we have this lipolysyl arm, right? And this is connected to E2. And on this arm, we have amino acid to begin with. So here is lysine. And we have this extra bit that is connected to lysine. All in all, we're looking at 14 angstroms. Just in case you don't remember what an angstrom is, an angstrom is the distance of 1 carbon-hydrogen covalent bond. That's 1 angstrom, or 0.1 nanometers. That's also an angstrom. So it's 14 angstroms long, and it's very flexible. You can see that other than this C double bond O, everything here is a single bond, carbon, 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 right? And so this means that this is highly flexible, and it's like a giant tether. And it's going to be connected to a proline and alanine-rich segment. And it functions like a giant arm where, this E2 will go with its arm and take the pyruvate from E1, then it will extend to E3. So if you can imagine, like each one of these E2s, I guess I'll do it in here. Each one of these green spheres has like a little arm that reaches out to it. and it can go and interact with any E1 molecule that's near it. And then it can go interact with E3. That's why it's saying each E1 can acetylate numerous E2s. They're all in very close contact. And E3 can go and interact with various E2s as well. So not only does this help facilitate the movement of substrates to make sure they go to the right enzymes, but it also makes the process more efficient by making this complex. Each enzyme can interact with multiple copies, right? Each E2 interacts with multiple copies of E1 and E3, which is different than a lot of enzymes when we look at mechanisms like for glycolysis, right? Usually when you create something, it just goes on to the next enzyme, and it's in a chain. Here, I have a lot of options, so I can do this very fast. So any questions about our PDH complex or our lipolysyl arm or the way PDH is organized or anything at all? Right, if you are still typing on, please continue to do so. Otherwise, I'll move on. So here I just have a little bit of refresher review for us since it's been since Thursday since we talked about PDH. So I want you to go back and do a little studying right now. And I have a poll for this. If I can get the right question up. I want you to tell me for E1, what coenzyme do we need for E1 to work? So if you're not sure, use this time to look back at your mechanism. And again, this is just another way to reinforce our learning of this by constantly going back to old information. Do about 10 more seconds on this. All right, and it looks like, yep, very good, everybody. That is TPP, thymine pyrophosphate, also known as TPP, is what starts off our mechanism of E1. So E2, that contains a lipol group that is attached to what? We just talked about this. So make sure you go back to this when studying for the exam, but since we just talked about that, that's a lysine. That's why it gets lipolysine. That's why we call it that. And then question 6. So pyruvate plus NAD plus coenzyme ASH goes to acetyl-CoA plus NADH plus H plus carbon dioxide. I have 3 options, and I know everybody loves questions like this. Tell me, of these 3 options, what do you think is the correct answer? So take a minute, think about those, look at those options, go back to our mechanism, relook at the mechanism, and see which one you agree with. do about 30 more seconds and then we'll go and look at the mechanism together. All right. So this one, this question is definitely one of the more tough ones, where it is definitely testing your understanding of PDH, right? So let's take a look at this. So we're going pyruvate going to acetyl-CoA, okay? This enzyme that catalyzes this reaction contains a pyridoxal phosphate prosthetic group. So let's go back to our mechanism here. And the reaction, right, goes to pyruvate to acetyl-CoA, right? That's basically what we're doing here. So we're looking at the whole mechanism though, because we also have our NADH. So this question is the whole PDH mechanism written in just its chemical form. And the substrates, or sorry, the coenzymes we have are TPP. Then we have a lipoamide. And we didn't really talk about this, but this is a dihydrolipoyl. Right. And so we don't really have a pyridoxal group in here. So that doesn't appear anywhere, a pyridoxal phosphate. So that's not true. The only really cofactors or prosthetic groups we care about are TPP, right? And lipolysine, right? Those are the only two like real prosthetic groups for PDH. It doesn't really contain anything else. This reaction is oxidative decarboxylation. Well, we do have decarboxylation. That means you are making a CO2. And it is a redox reaction. Remember, whenever you see NAD and NADH, that means a redox reaction. So number 2, yeah, that's true. This enzyme is activated by high concentrations of ATP. And if we go back to it, we didn't really talk about, how this is activated, right? So throughout our discussion, I know the writing's there, so I apologize for this. Let me clear that. Should have done that before I went back. Yeah, ATP doesn't actually turn on PDH itself. We'll talk about later today what actually does turn it off, but ATP itself doesn't activate PDH. So it's not that. So the answer is 2 only. Oxidative decarboxylation. Right, so any questions about PDH now that you went back and looked at the mechanism, refreshed yourself on the mechanism, anything at all? All right, well, if you do, please continue to type. I do have one more question, question 7. We'll skip this because PDC itself is irreversible. So PDC is irreversible. However, this question is really designed to see what your thinking is of looking at each individual steps, and based on what we learned through other mechanisms, which one would you say could be reversible? The answer to that is most likely 4. because all you're doing is you're breaking and reforming disulfide bonds. So step one, you're breaking off a CO2, so that's irreversible. We've seen that before. Step two, you're using the energy of breaking a disulfide bond to move carbons. Usually, since this is the energy going in, to do that reverse, you would have to put some energy in. Three, same thing where you are using coenzyme A as your energy source to move carbon. So you can't break that bond without putting energy in. So four, you're just switching disulfide bonds. So that one's probably the closest to a delta G of 0. 5, you're using NAD plus NADH. But that is really designed to be a big thought experiment for everybody if we had time. Unfortunately, I need to keep on going. But just keep in mind, PDC itself is irreversible. And I won't ask you like this question on a test or anything. So if you just want to put like a big X next to it, yeah, this is one you can skip in your studying. And I believe that's the end of this PowerPoint. So give me a minute and I will pop up the next PowerPoint for us. 3.2 PowerPoint. So let me get that loaded. All right. So today, the rest of today, we're going to talk about the citric acid cycle proper, right? So Thursday, in the beginning of today, we talked about the activation step of how to go to pyruvate to acetyl-CoA. The rest of the time today, we're going to talk about doing the whole cycle of the citric acid cycle. We already mentioned the basics of it before, how we're making NADH, FADH2, GTP, getting rid of carbon dioxide. To note, the carbon dioxide that you lose is from oxaloacetate. It's not from acetyl-CoA. So the picture actually tries to show you that by saying this carbon starred, and you lose it here at this starred carbon dioxide. And the other carbon dioxide we lose is this one, because it becomes C double bond O, and you can see it gets lost here at isocitrate dehydrogenase. A lot of these molecules that we see in the citric acid cycle are going to keep popping up again and again when we talk about metabolism, because they are used for a lot of different purposes here. But let's get into the actual mechanisms here. So this is going to be very similar to glycolysis in the way that we're going to look at this. So our first step of the citric acid cycle is we're going to take our acetyl-CoA that we just made from PDH. Right, so this is coming from PDH. And we're going to combine that with oxaloacetate, which is actually the last step of the citric acid cycle. The last step of the citric acid cycle, we make oxaloacetate. In the first step, we just combined a 2-carbon compound with a 4-carbon compound to make our 6-carbon citrate. For this, don't worry about mechanism. Even though I have the mechanism here, the big mechanism thing we're going to focus on right now is PDH. So not to overburden you with just pure memorization stuff, I'm not going to ask you questions about the mechanism of citrate synthase, but you should know what's happening here. And what's happening is that you take acetyl-CoA, and you combine it to oxaloacetate to make citrate. Acid-base catalysis, that's what's happening here. And it is extremely negative. It is negative 31.5. And the reason why it's so negative, and we talked about this at the very beginning of this course, but it wasn't really emphasized all that much, is because this coenzyme A. 1 second, I don't want to sneeze in your. So this coenzyme A, this is a very highly charged, or sorry, very highly high energy molecule, energy rich. That's the word I was going for. It's an energy rich molecule. It's kind of similar to ATP. Similar, but not quite. The way that coenzyme A works is that you attach it to a molecule, and that molecule becomes very high energy. And it's the same idea of taking ADP and attaching a phosphate to ATP. You make a very high energy bond by doing that you can use later on for different processes. Coenzyme A, same idea. Very high energy molecule that you put on something. Once you break the coenzyme A off, that's a lot of energy. So we're using our coenzyme A as our energy source here to make a carbon-carbon bond and to combine 2 carbons with 4 carbons to make our citrate molecule. Right, so any questions about our first reaction of citrate formation from oxaloacetate and acetyl-CoA? All right. So once we have citrate, once we have made our new 6-carbon compound, we just need to change it to isocitrate. This is a isomerization. So remember, an isomerization it's similar to a mutase in that all you're doing is moving a functional group. So here, all we're doing is we're taking OH from this carbon and moving it 1 down to make our isocitrate. This does have a intermediate, which is stable. You can actually measure the amount of this intermediate called cis-aconitase. And this is simply a dehydration-hydration reaction. So that's how we're moving the OH. We're just taking water off, putting water back on. The actual enzyme that does this, aconitase, the way we're moving this water is that we have what's called a iron sulfur cluster, right? It's just a bunch of iron and sulfur molecules all combined together. So 4 irons, 4 sulfurs. We're not going to get into the exact details of iron sulfur clusters at this minute. But once we get to oxidative phosphorylation, we're going to see iron sulfur clusters a lot. So this will be like our first introduction to them. And what they're doing here is that they're just helping move the OH groups around. They're the one facilitating the removal of water in the addition of water. So they are really the powerhouses in doing these processes. As I say here, normally when you see an iron sulfur complex in a protein, that tells you, okay, we're doing redox reactions. I should expect to see an NADH or an FADH2 somewhere. Here, that's actually an exception to that. We're not doing redox reactions. We're just moving OHs around. So that's our second reaction. Citrate going to isocitrate with our cis aconitase intermediate. Still at 6 carbons though. Remember, the goal is to lose 2 carbons. The goal is to make oxaloacetate, and that's what we're setting up to do. Now, just take a brief respite from actual look on this mechanisms, and let's look at energies. Energies are very important here. If we look at aconitase, the delta G for aconitase to move those OHs around is a positive 5-ish. However, inside the cell, this is basically 0. And this is a concept we talked about in glycolysis already. Why inside the cell is this like neutral? Why isn't it a positive value anymore? What is driving this reaction forward again? Now there's only a few people here today, but do any of you want to share why this reaction goes forward? Coupling, yes. If there's any benefit to study metabolism, it's that the core concepts are repeated over and over and over again. True, the actual molecules change, but it's just the same idea with different players, right? You can see the next enzyme, isocitrate dehydrogenase, very negative. So what happens for aconitase is when we do the reaction, it is immediately used by isocitrate dehydrogenase. The concentration of products are kept low. And due to equilibrium, if you take away products, you're going to shift the equilibrium to make more products. Le Chatelier's principle, Gen Chem 2. And this reaction is coupled to a different reaction. And really, the whole citric acid cycle, since it's a circle, all the reactions are coupled to each other because they all work together. Yeah, so that's the answer, coupling. Like how in Biochem 1, if you didn't know the answer to something, put hydrogen bonds, you have a pretty good chance you have a decent chance at least getting some points. You don't know the answer to something, coupling. That might be the answer. It seems to be the answer for a lot of things. All right, but back to our next reaction. So we have isocitrate that is made by aconitase. Now we are doing our first decarboxylation reaction. Isocitrate dehydrogenase, all right? And I've talked about this before. Dehydrogenase. Whenever you see the word dehydrogenase in an enzyme, that's your cue that we're doing redox reaction. So we should be looking for NADs or FADs to be part of the mechanism. And that's what's happening in the first step here. We have NAD plus going to NADH, right? So we are taking off the hydrogens from this carbon to make a C double bond O. Now, just looking at oxalosuccinate, which is one of our intermediates, it looks like a very unfavorable molecule. You have a negative oxygen, and a very polar carbonyl group. So this is very negative itself. So you just have a big area of negative charge all next to each other. So normally this would not want to form. So we need a cofactor in the form of manganese or magnesium can also fit. This will help to balance the negative charge of our oxalosuccinate, a concept that we have talked about in glycolysis especially when we're looking at phosphates being ATP being created or destroyed. Those enzymes always had magnesium to balance the negative charge. Here, same idea. Okay, so the next step in our mechanism is that we lose our carbon dioxide, and we have this very short-lived molecule. So you can see it doesn't even have a name next to it. That means it's like so short-lived you can't like measure it. Where we have a double bond, oxygen, oxygen. Acid comes in, removes the double bond by attaching a hydrogen to the carbon. The double bond reforms, and we get alpha-ketoglutarate, or I'm going to talk about it, I'm going to call it AKG a lot. Alpha-ketoglutarate, we will talk about a lot in this class moving forward, because it is one of those molecules that is seen a lot in different pathways, especially the ammonia, how to get rid of urea pathway. Alpha-ketoglutarate is a big player there. And it's also a starting material for a lot of the things that your cells make is alpha-ketoglutarate. But we'll focus on that on a later lecture. Right now, we're just looking at isocitrate, makes NADH, loses a carbon dioxide to form alpha-ketoglutarate. Remember, these two carbons were from our acetyl-CoA. So we're losing the carbon dioxide from our original molecule, not acetyl-CoA. So any questions about our isocitrate dehydrogenase reaction where we lose our first carbon dioxide? All right, let's move on then. Next reaction is we're going to take the AKG we just made and make succinyl coenzyme A. And this is done by alpha-ketoglutarate dehydrogenase. And like I just said on the previous slide, we're doing redox. So there's our NAD. And what we're doing is we're removing our other carbon dioxide at this point. So we're going back to 4-carbon compound. So we have lost our 2 carbons already that we have to lose. Remember, not from acetyl-CoA though. And we also have our friend coenzyme A. In case you're wondering, this SH, that's the reactive part. Coenzyme A is like a huge molecule. The only thing we really care about in terms of energy is this sulfur bond. So that's why you'll often see it as CoAsh. It's just showing you, oh, this sulfur is what we care about, right? And going from AKG to succinyl-CoA, it is not as simple as this. There is an alpha-ketoglutarate dehydrogenase complex. It's basically the same idea as PDH. And since we went through PDH, I'm not going to go through alpha-ketoglutarate dehydrogenase, but you have 3 enzymes. You have your lipoyl arm that's moving around, and you basically do the same thing, just with a different molecule. But that is our next reaction. Alpha-ketoglutarate dehydrogenase, you're making succinyl coenzyme A, making another high energy bond that we're going to use. getting rid of CO2 and making more NADH. And I don't think I mentioned this today, but remember NADH, that's basically electrons. Anytime you see NADH or FADH2 being formed, all that means on a chemical sense is that we have transferred electrons in the form of a hydrogen. So we are moving 2 electrons around, so 2 electrons. And we use that as currency for oxidative phosphorylation, which we'll talk about in a few weeks here. So our next reaction, and to save you time, again, there's a lot of memorization in this class. So I have the mechanism here. No need to memorize this. Like I said, we're going to focus on PDH. That was our big thing so far. So no one need to memorize this mechanism. But the next reaction is succinyl-CoA synthetase. This is a weird enzyme name because in the way we learn it, we are not making succinyl-CoA, we're making succinate. The reason why this is called succinyl-CoA synthetase is that this reaction is completely reversible. So it is possible to go from succinate to succinyl-CoA. So my guess is whoever discovered this discovered the reverse reaction first, but in the citric acid cycle, we are mainly focused on the forward direction. For the citric acid cycle, this is the first time that we are doing substrate level phosphorylation. We talked about this in glycolysis, but substrate level phosphorylation means we're adding a phosphate directly to a high energy compound. We're mammals, so we make GTP. Plants and bacteria make ATP. Why are they different? I do not know. It's just that we have slightly different versions of the enzymes. But again, I talked about this on Thursday when we first talked about the cycle. This is just another form of energy currency. It's similar like to the euro, where it's just used in different enzymes that ATP can't be used in. 3 steps to this. Again, I think for the sake of time, we will just skip that because I'm not going to have you memorize that. But know that what's important to know is that we're taking succinyl-CoA, losing the CoA to make succinate, and we're using that energy of, remember, because this is high energy bond, we're using the high energy coenzyme A bond to attach a phosphate. So GDP goes to GTP. So we're making GTP here. And the important residue is histidine. Histidine's an all-star when it comes to mechanisms. It does everything. Here, histidine is simply a phosphate holder for us, taking phosphate from our intermediate and attaching it onto GTP. And so if we look at where we've come so far for the citric acid cycle, 1 acetyl equivalent. So what I mean by that is that we've lost 2 carbons. Acetyl group has 2 carbons onto it. But again, I'll reiterate this one more time. The carbons we lose in CO2 is not from acetyl-CoA. But anyways, we've created 2 carbon dioxides. We have created 2 NADHs and 1 GTP. The only thing that's really left to do for us to complete this cycle is that we have to change our succinate into oxaloacetate, because that is what started the cycle, and that's what combines with acetyl-CoA. So that's what we're going to be working towards now. How to get this succinate to look like oxaloacetate. So let's move on to our next reaction then. Okay, reaction 6 is that it's a dehydrogenase. So hopefully, since this is the third time I told you, you know what to look for. Look, you have it right there. This dehydrogenase is different than the other two we have looked at today because the FAD, which is doing our redox, is connected to the enzyme. That's what the E means. So this is actually bound to the enzyme. The other molecules we looked at, they weren't. They were just floating in your mitochondria, and you just grab it, use it, and then release it. This one is attached to your enzyme. And the reason for that is because succinate dehydrogenase is actually an enzyme in oxidative phosphorylation. So it's serving double duty for us in terms of how we're going to learn about it. So we're just going to focus on its role in citric acid cycle. And then when we talk about oxidative phosphorylation, we're going to talk about this enzyme again. And I'll say, see, remember this from the citric acid cycle? Here it is in action. But here we're taking succinate, removing 2 hydrogens, and making the stereospecific fumarate, where you have a C double bond O on each side. This is inhibited by the molecule malonate. If you squint, malonate kind of looks like fumarate. You just miss 1 carbon here. So this malonate, if you have any around, it will completely stop this reaction from happening. Like I said, the FAD is attached to our enzyme via histidine. You make FADH2. To get this enzyme to do another reaction, you have to lose these protons, you have to lose these electrons, and so that will be shown how that's done in the electron transport chain. But we'll get to that when we talk about oxidative phosphorylation. All right, so we are now at fumarate. Our next step is to make malate. And this is done by the enzyme fumarase. And the difference between fumarate and malate is that you simply get an OH group on one of your carbons. Here we have a very interesting transition state, a carb anion, which we have a negatively charged carbon. And if you remember from organic, that is very unfavorable. So this is very short-lived. But generally what happens is that you have hydration to add an OH group that breaks the double bond. And so 1 carbon has 4 bonds, so that's happy. But 1 carbon only has 3 bonds and a lone pair of electrons. That's what's making it negatively charged. But with a carb anion, you can easily add a proton to it, which is being shown here, and that will make us our malate. Again, not too much to say about this actual reaction. Relatively straightforward. Fumarate, malate, OH plus H. All right, and now our last step. We've finally made it back to oxaloacetate. We're going to take the malate and we use malate dehydrogenase. Okay, this is like playing Blue's Clues now. Many of you have watched that show before. Where is the NAD? There it is. So dehydrogenase, look, we have a redox reaction. And we are making NADH and we are losing our 2 hydrogens to make our oxaloacetate. This reaction depends on if you have NAD plus or not. If you do not have any NAD plus, you will not continue the citric acid cycle. This is extremely unfavorable reaction, right? Plus 30. However, the first reaction is minus 30, 31. So just doing some math, right? 29.7 minus 31.5 equals negative something something. What is that, 1.8? So overall, when combined, with acetyl-CoA, since these two reactions are coupled, overall, spontaneous. That is not true if NAD plus is low, because the lower this gets, the more unfavorable this reaction becomes. And even if you have acetyl-CoA available, this reaction won't run unless you have also NAD plus available. So this is a good way for regulation, right? NADH is basically energy. I put it in quotes there because it actually has to go through oxidative phosphorylation to become energy. But if you have a lot of NADH, you have a lot of energy that's waiting to be cashed in. The citric acid cycle, the whole job of the citric acid cycle really is to make energy. So if you don't have a lot of NAD plus, that means you must have a lot of NADH. You already have a lot of energy waiting to be created. Therefore, don't run the citric acid cycle anymore. This reaction will stop. And once that stops, intermediates pile up. These intermediates can be used for a lot of different things. So that's what's going to happen to all the carbon complexes. We're going to use it to build other things rather than destroying them as carbon dioxide to make energy. So that's the logic there for NAD+. And that also ends the citric acid cycle, looking at each reaction one by one. So after that, any questions people might have about anything we have talked about up to this point? All right. So overall, what has happened? What have we spent the last month learning about? And that says GTP down here. That's what's coming off. So we looked at how glucose can go to 2-pyruvate. When we do that, we get 2 NADH. Each NADH is roughly 2.5 ATP. So we make 5 ATP. And we have made 2 ATP from substrate phosphorylation. From pyruvate going to acetyl coenzyme A, remember that's the PDH complex. For every 1 glucose, we get 2 pyruvates. Therefore, we do PDH twice. We get 2 NADH from that, 5 ATP. Then the acetyl-CoA, the citric acid cycle, that's where we really get the majority of our energy, right? So for every glucose, you run the cycle twice. So you get 6 NADHs, 2 FADHs, which are equivalent to 1.5 ATP, and you get 2 GTPs. And when we talk about oxidative phosphorylation, we'll actually go over why NADH gets more ATPs than FADH. So we'll cover that. But here in the total citric acid cycle, not even considering PDH, for one glucose, you are looking at 20 ATP. While from glycolysis in PDH, you get 12 ATP. Overall, if one molecule of glucose gets broken down all the way, that is 32 ATP. All those electrons, the NADHs and FADHs, that's going to the electron transport chain. Since we are creating a lot of energy and we are using a lot of resources, this whole process, and we've already looked at it, but this whole process is very highly regulated. It's regulated by amount of substrates. It's regulated whether you need intermediates for different things. It's regulated for if you actually need ATP. So everything here, very highly regulated, but that is our total energy gain so far from this class that we've learned about. All right, so I've been talking for a long time, so let's do some questions to get you re-engaged in case you fell asleep over there. Now, thinking about the citric acid cycle, we've been mainly focusing on how it's done in the majority of organisms. However, not every organism runs it the same way. So some microorganisms run it kind of incomplete, where you create alpha-ketoglutarate, and then instead of going to succinyl-CoA, you make what's called a Succinal Semi-Aldehyde, as shown here. So this is the reaction some organisms do. From there, you can make succinate, no problem. So if we were looking at this reaction, comparing the Succinate Semi-Aldehyde form to the form we just learned about, how would our ATP be different? Like, what would change between this microorganism pathway compared to the pathway we just talked about in terms of ATP or in terms of our cofactors? So what's the difference? And I guess another way to ask this is, in the way we just learned, what do we get out when we go the normal route of succinyl coenzyme A to succinate? Anyone have any ideas about the difference in terms of energy between these two similar ideas? Going to succinate semi-aldehyde versus going to succinate directly. Well, let's take a look at this, right? When normally in mammals and most organisms, we go of alpha-ketoglutarate to succinyl coenzyme A. From that, we get an NADH. So I'm going to put the mammal pathway, MAM, 1 NADH. and other microorganisms. So they go from AKG to semi-aldehyde. They don't get anything. They just lose carbon dioxide. Right, so from succinyl coenzyme A to succinate, that's when we're gaining 1 GTP. So right now we're at succinate, and that's the only difference between the two pathways is this succinyl-CoA middle step. Here, from succinal aldehyde to succinate, we create NADH. So just comparing the two paths, the only thing that's different is you are not creating a GTP. So that's going to be the one loss here. You lose 1 GTP or, depending on the organism, 1 ATP. And that's why it's kind of called like an incomplete citric acid cycle. These organisms aren't getting as much energy out of the citric acid cycle as we are, because they just have a different enzyme there to make our succinate semi-aldehyde. So any questions about that idea or what that question was asking there? Yep, 2 reactions to get NADH and CoA versus, well, in mammals, it is still 2 reactions. It's just in one reaction, you get NADH, in one reaction, you get GTP. But yeah, you're right, 2 reactions to separate CO2 and NADH for those organisms. Yeah. So let's talk about regulation of the citric acid cycle. And the first part of regulation is PDH, our pyruvate dehydrogenase complex. Again, we said PDH is irreversible. So once a pyruvate enters into PDH, you are going to be made into acetyl-CoA. There's no going back. So it makes sense, that this would be highly regulated as it's all downhill from here. 2 different ways to do this. One, we can inhibit PDH by the amount of NADH in acetyl-CoA. Again, remember, these things mean high energy. NADH makes ATP by the electron transport chain. I'm going to say that's EC here. Acetyl-CoA, it's fed into the citric acid cycle itself to make ATP and more NADH. So if you have levels, very high levels of NADH and acetyl-CoA, it really doesn't make sense to run the citric acid cycle more because you're already maxed out on the amount of energy you have. Therefore, the logic is use that pyruvate to do other processes. Maybe make some glucose out of it through gluconeogenesis. Maybe make some nucleotides by doing the phosphate pentose pathway. We can do a lot of other stuff with that pyruvate than just straight up and make energy. And the reason how this works is that this NADH will bind where the NAD plus binds, and acetyl-CoA will bind where normally coenzyme A binds in PDH. And so enzyme 2 and enzyme 3 just won't go forward anymore. They will go backwards. So this machinery is basically just stopped. It will no longer go forward for you. So no pyruvate can enter into that enzyme complex. So that's called product inhibition. And covalent modification, phosphorylation, dephosphorylation. We talked about this a bunch already. Again, this is something those of you going to any type of biomedical biofield, you're just going to get beat over the head with this. Phosphorylation controls a lot of stuff. Here we are phosphorylating and dephosphorylating E1, the first enzyme in PDH. And it's controlled by 2 different enzymes. Pyruvate dehydrogenase phosphatase. Remember, a phosphatase usually removes a phosphate. And pyruvate dehydrogenase kinase. A kinase normally adds a phosphate. So NADH and acetyl-CoA, not only do they bind PDH, they activate pyruvate dehydrogenase kinase. Once that's turned on, ATP will be used to add the phosphate to a serine residue. Remember, serine has the side chain as OH, so serine's often an amino acid that will get phosphorylated. Once phosphorylated, E1 shut off, and you can't run PDH. Insulin, on the other hand, insulin will go turn on the dehydrogenase phosphatase, which will remove the phosphate, making E1 active. Remember, insulin is a sign for having a lot of glucose in your blood. So if you just ate a meal, you're going to have insulin flying around, and it's going to tell your body, okay, we have glucose coming in, get this citric acid cycle working so we can start harvesting energy. What can inhibit the kinase? So if you've never seen like a symbol like this before, when looking at different mechanisms, when you have a straight line up and down, that means inhibit. Those of you taking the MCAT, you should get used to that symbol, because there's questions on the MCAT where they just show you novel pathways and ask you to figure it out. So what can inhibit the kinase? High levels of pyruvate, high levels of ADP, and calcium can inhibit the process as well. So that's regulation of PDH. Any questions about that so far? All right. Now, talking about the actual citric acid cycle itself, let's take some of the lessons we learned from glycolysis and apply it to regulation. So to be an enzyme that controls flux, you have to operate far from equilibrium. And we are talking about normal cellular concentrations. So remember, we care about delta G here. Delta G not prime is just standard conditions we use to compare different enzymes. What we really care about when we talk about regulation is just delta G itself. And there are 3 enzymes that are not near equilibrium that can be regulated, right? Enzyme 1, enzyme 3, enzyme 4. So citrate, synthase, isocitrate dehydrogenase, and alpha-ketoglutarate dehydrogenase. So these will determine flux for us. And flux here, 3 different mechanisms we use for flux. How much substrate is available? Do you already have a lot of products around? So product inhibition. And feedback, competitive feedback by various intermediates. And we're going to get into each one of these on its own. First, let's talk about NADH. We mentioned this already when it came to malate to oxaloacetate, but NADH is one of the most critical regulators of the citric acid cycle. And let's look at this oxaloacetate to malate reaction again using our equilibrium constant K. So remember, K, back from general chemistry, products over reactants. Right. So let's think about this. Let's say you are exercising and your muscles are working. When your muscles are working, the amount of O2 you are breathing increases. And what you're trying to do is you're increasing oxygen to run oxidative phosphorylation. That means oxphosphorylation also increases because you're trying to make ATP. To run oxidative phosphorylation, you need electrons, and you make electrons, or you give electrons to oxidative phosphorylation via NADH. So NADH gives 2 electrons to this process, and you create NAD plus now. So the idea here is when you breathe more, when you're exercising, the amount of NADH goes down and the amount of NAD plus goes up. What this does is that it propels malate to be made into oxaloacetate, which will then turn on this first reaction. So this will turn on citrate synthase and create our citric for us. So that is one way NADH can move the reaction forward. Let's look at the opposite though. What if you do have high levels of NADH? That's what's being shown in this image. So NADH is at the center of our circle because it's just saying it's very important. What these red lines are showing, they're showing inhibition. So what this is showing is that if you have high levels of NADH, this will go and inhibit PDH. This will go and inhibit citrate synthase. This will go and inhibit isocitrate dehydrogenase. This will go and inhibit alpha-ketoglutarate dehydrogenase. So our 3 main enzymes that we talked about, they are inhibited by high levels of NADH. Because as we talked about, NADH means energy. So if you have a lot of NADH, don't run the citric acid cycle. Right, so before we move on, there were any questions from the information presented here on this slide, or any of the little tangents I went on? Right, so just looking at this plot a little bit further, we can also see that there's other intermediates that inhibit stuff. So succinyl coenzyme A will go and inhibit alpha-ketoglutarate dehydrogenase. So that's what this is also inhibited by. So we'll here, so that's succinyl coenzyme A. And it also goes and inhibits citrate synthase. So that's also inhibited there. So this is competitive feedback inhibition, when succinyl-CoA is going to inhibit the first molecule. And you have ATP going to inhibit isocitrate dehydrogenase. So ATP also inhibits this. What turns on the citric acid cycle? Calcium. Calcium's a big signal saying, okay, let's go and turn on the citric acid cycle. So it turns on PDH for us. Also turns on isocitrate dehydrogenase. Also turns on alpha-ketoglutarate dehydrogenase. And also ADP. So ADP, your low energy, turns on isocitrate dehydrogenase for us. So that's what that image in the bottom right is showing. And that's how you follow those lines when that's what the red and green means. What's processes turning on, processes are turning off. All right, and I have a bunch of questions here, but I think we're going to use those questions as review on Thursday so we can remember what we talked about today. So with that, that's where I'll end our discussion for today. If you have any questions, as always, do not hesitate. Let me know, email, whatever. I'll put a homework up to reinforce the concepts that we learned today. Otherwise, that's all I have for you. And look for that test walkthrough. If not today, definitely by tomorrow. And I will see people later. Have a good Tuesday, everybody.